So good evening, everyone. The Fabry Australia Committee welcomes you to this information session regarding upcoming clinical trial opportunities. My name is Sheridan Campbell, and for those of you I've not yet met, I'm the Chair of Fabry Australia. I'm joining you this evening from Yorta Yorta country in northern Victoria. I pay my respects to the Yorta Yorta elders, past, present and emerging, and I invite you to consider the traditional owners of the land where you're joining us from this evening. Thank you to the Fabry Australia Medical Advisory Committee for making themselves available for this virtual event. We're fortunate to have Dr. Kathy Nichols from Melbourne, Dr. Mark Thomas from Perth, Dr. Charles Denaro from Brisbane, and Dr. Michael Chen from Sydney on the conference with us this evening. Firstly, we'll hear a presentation from Dr. Nichols, followed by an opportunity to ask questions of the specialist panel via the chat function. Please note, we can only address questions this evening relating to the clinical trial programs. If you need to ask a personal medical question, it's important you follow up with your treating specialist. Dr. Kathy Nichols is a kidney specialist at the Royal Melbourne Hospital. She coordinates Fabry-related clinical care alongside her team for over 100 Victorian patients. The Royal Melbourne Hospital unit has treated Fabry patients with enzyme replacement therapy since 2000. And continues to participate in clinical trials for emerging therapies. Royal Melbourne Hospital is just one of the trial sites for upcoming trials. So without further ado, I'll pass over to you, Kathy. Thanks, Kathy.
Okay. Thanks, Sheridan. Well, thanks Sheridan and Diane and Fabry Australia for the opportunity to get together and have a bit of a chat about the upcoming gene therapy programs. We've been waiting a long time to have access to gene therapy for Fabry, so hopefully this will help introduce the concept and some of the specifics of the program. Anyway, that video is just a general introduction to gene therapy. It's not specifically Fabry anyway, but it's freely available on the website, which I can provide. So the gene problem in Fabry disease is one that I'm sure most people on the call are quite familiar with. The gene is GLA. Its gene product, the enzyme that's deficient in Fabry disease is alpha-galactosidase. And without that sufficient enzyme, you get a substrate buildup in the cells of sphingolipid stuff called GL3 or GB3 and lyso-GB3, which causes the tissue damage. So there's many different mutations in the GLA gene that can produce Fabry disease by the change in the gene producing an abnormal protein or sometimes no protein at all. And as you know, the general gold standard therapy for over 20 years has been enzyme replacement therapy to introduce human enzyme intravenously every couple of weeks. And that's been a life-changing therapy for many people with Fabry disease over 2 decades now. It has got a downside. The treatment for Fabry disease currently is not perfect. Enzyme replacement therapy is not a cure, it just slows the progression. It does improve lifespan and improve quality of life, but there is a treatment burden to fortnightly infusions, and people tend to run out of enzyme at the end of the second week of the end of the 2-week cycle. Enzyme has got some limitations in terms of how many of our tissues actually get distributed enzyme from intravenous infusions. And there's a problem particularly for people who don't make any enzyme at all, in that you can form antibodies against it, so that the enzyme replacement therapy actually acts as a vaccination. So can gene therapy help us and can it be a better treatment? Well, hopefully it can, and hopefully it's one treatment that's potentially more effective and overcomes some of the downsides of enzyme replacement therapy. It is designed to allow continuous production of the enzyme. And Fabry disease seems to be, at least theoretically, is particularly amenable to gene therapy, because you don't need to put the gene into every single cell. You can put it into a reservoir of cells, and then those cells can secrete the enzyme into the blood and distribute it around the body that way. And we may be able to treat a difficult group of people who are immunized against enzyme. They have antibodies to alpha-galactosidase, the Fabry enzyme. And at least in mice, gene therapy is has been incredibly successful at boosting enzyme levels. And it doesn't seem like if you overshoot and get a lot of extra enzyme, it doesn't seem to matter. So there are 2 gene therapy programs for Fabry disease opening up in Australia. There are 2 different companies and 2 different products. The 2 companies are Sangamo and 4D Molecular Therapy, and they use 2 different viral vectors. The vector is the sort of packaging that you could put the gene inside and allow it to get into the cells. Because if you just put a gene in by itself into a vein, into the body, it'll just get destroyed before it gets actually into the cells and starts to make enzyme. So the 2 different programs have many similarities. So there's a screening phase where, depending on whether you're male or female, depending on the severity of Fabry disease, and depending on the particular mutation that has caused Fabry disease in your family, and whether or not you're on enzyme replacement therapy or not on enzyme replacement therapy or on chaperone therapy, you can enter a screening process, and then if that all looks okay according to the trial protocols, be treated with a single dose of the gene therapy product. That is followed then by evaluation and follow-up that's quite intensive, at least over the following 12 months, and effectively it'll be a long-term follow-up. So if we look at the each of the programs individually, for the Sangamo program at the moment, it's just for males, although they will be accepting under a variation of the trial protocol that's coming soon, females as well. Usually most of the patients accepted into this program will need to have classic mutation and not be on enzyme replacement therapy. And there are some other clinical criteria that are required before people are eligible. The 4D program, 4D Molecular Therapies, is a very interesting product, and we will talk a little bit about that. It is a little bit more permissive in terms of who's eligible for the program. It's at the moment just for males, although it will be opening to females very soon. The mutations can be either classic or non-classic. People can either be on ERT or on no ERT. And there are some other clinical criteria that need to be met before being eligible for screening. And if that all works out okay, then you can get the dose and again go into the follow-up program. So it's a bit complicated, but if we kind of draw a map of starting out whether an individual person is on ERT or not on ERT currently, and whether they're male or female, then males on ERT and soon females can go into the 4D. if you're not on ERT, you may have a choice, at least in centres where both programs are available, to go into either program. We're going to need to wait on females, but males are ready to go in terms of the 4D program. People on megalostat can also be considered, although the need to cease the megalostat when they're being treated with the gene therapy. And there are maybe some restrictions involved. If you're already in another trial, that investigational drug will need to be ceased a month or 4 weeks before the screening process. So there's sort of, conditions that need to be met to go into these studies, but they are available for males now and will be also available for females very soon. The Sangamo gene therapy product is wrapped up in a viral, what they call a capsid, which acts sort of like an Amazon drone, really, to deliver the gene inside it, which is packaged up in there in a product called ST920, and that is infused intravenously into the patient and goes to the patient's liver. And from there, it secretes enzyme into the rest of into the circulation, so that you have a continuous level of enzyme in the system. It's early days. This is the study. This is the first 6 patients treated with the Sangamo product as up till February this year, and this has been presented and published. So this is up the graph, or the block is vertically is the circulating enzyme level in each of the first 6 patients. The dotted line is the normal level of enzyme, and the blue block is the range of normal enzyme. And you can see that after infusion of the gene therapy product, patients had quite a lot of enzyme in the system. And that actually has happened quite early on after the product is given. The 4D product is a very interesting product, and in that it's very similar to that Sangamo in the sense that the gene is obviously the same. The vector is similar. It's another adeno-associated viral vector, or AAV. All the viral replication genes are taken out, but the capsid, or the packaging outside, allows this product to go not only to the liver and to sit there long term, but also to get into the heart cells. And that may be particularly useful because enzyme in the blood tends not to get into heart very, very effectively. And again, it's early days. These are the first data from the first couple of patients. So again, enzyme activity up on the y-axis, and patient 1 and patient 3 are shown when after the dosing, the enzyme level went up very, very high and very fast. And over time, over 5 to 7 months or so, it's actually stayed up. And this has been also sustained in subsequent patients who've been treated with the product as well. So they both seem to be successful in terms of putting enzyme into getting the gene in, and then subsequently the gene making the enzyme. The companies, will it work? Well, it's early days. It looks pretty good so far. That's about all we can say, really, at this stage. We need to be able to show, and the FDA will require the companies to show, that the circulating and white cell enzyme levels are not only adequate, but that they're sustained over time, and that GB3, is cleared from the tissues after the product is given. So to do that, patients are going to need to undergo testing at baseline and after throughout the first year. Now, most of those tests are pretty similar. Clinical assessment, like most people are with Fabry, are pretty much used to. And blood tests are pretty much the same for both programs. Skin biopsy to show that the GB3 is getting cleared from the skin cells. And the tissue biopsies at baseline and at 12 months, similar to the other Fabry clinical trials that have been done over the years. And they're a little bit different between the two programs. Sangamo are concentrating on the kidney, and 4D are concentrating on the heart. And we'll talk a little bit about heart biopsy because it sounds pretty terrible, being told that, oh, we want to biopsy your heart twice. But it's not as bad as it sounds. So the way that it's done is to put a skinny little wire with a contraption that actually pinches a little bit of tissue from the end of biopsy wire. And it could be it's put into a vein, either from any big vein really, from the arm or the groin or the neck where your big veins are up here. And it's fed down through the right side of the heart, doesn't go into your arteries, goes just down through the vein and goes, reaches the top of the heart here, sort of the bottom of the heart really, but we call it the apex. And it's a little biopsy is taken from the septum, which is the wall between the right and the left sides of the heart. Now, the size of the biopsy is 1 to 2 millimeters. So, and the thickness of the septum is normally about 10 millimeters or centimeter, and in Fabry disease can be, much more than that. Most people who will be in these programs have a septal thickness that's going to be at least 12 millimeters and probably closer to 14, 15. So you're not making a hole in the heart as such. You're taking a little bit of tissue from the septum of the heart, and then the whole thing just pulls out again. And it's not really an operation. It's like a central venous line. It's done by expert cardiologists. I'm not going anywhere near it. You'll be glad to hear. So, has gene therapy got a downside? Well, it's got some unknowns and some known downsides. One side effect of the vector itself, the packaging comes from a adeno-associated virus, and those viruses are made to go to the liver, and the liver can be pretty unhappy sometimes about meeting that AAV vector. And to overcome this, some patients may need to take some steroids to stop the inflammation or treat the inflammation in the liver. The vectors, you can actually get immunized to the virus, to the viral vector, to the capsid, and that might mean that, we don't know this yet, but it's theoretically possible that that might prevent you having another second go with the same vector. And for that reason, in both programs, there is some prevention treatment to prevent the antibody formation. And we can talk more about that. The 4D program has got a new protocol that will hopefully be better than steroids at preventing this antibody formation. How will the gene therapy compare with the currently available treatments with ERT or chaperone therapy, or with new treatments that are in development like long acting enzyme replacements or substrate reduction therapies, or even mRNA treatments? Well, we don't know. We haven't got any long-term data, and we're not going to have any until these programs become more widespread. So it's pretty exciting, but we want to proceed cautiously and very carefully and make sure that everybody's protected. The 4D program is open in Perth, in Melbourne and in Sydney. For other patients who would like to screen for these programs, the companies will provide travel and accommodation. So there's some uncertainty about it, and it's probably not for everybody, even all the people who qualify on paper and tick off all the entry criteria. People need to be suited not only physically, but it's not going to be an easy year. There's a lot of testing and a lot of visits, but it's more or less an investment for the future in terms of if it works and if it gets in there and is successful, then it should be, at least theoretically, a long-term treatment. Everybody who's interested should very much discuss it in depth with their own Fabry specialist. So here's the Fabry specialist. So we'll move on and take a lot of questions, or I hope there'll be a lot of questions. So Sheridan.
Thanks, Kathy. Yeah, thank you. I'm coming back in now. Thank you. Can everyone hear me? Sorry, I've had a few tech issues at my end, as per usual. Regional internet's not the best. I hate to say it. So thanks, Kathy, for your interesting and informative presentation. We've certainly learned a great deal. So I now invite Dr. Chan, Dr. Thomas and Dr. Denaro to join us for the panel discussion, where we'll attempt to answer as many questions as possible in our remaining time. Please type your questions in the chat box, questions that are relevant to the clinical trials program. So we've had a few already. So there was one question, is what are the age limits for eligibility? Do you have any idea?
Do you want me to take that?
Yeah, thanks, Kathy.
Yeah.
Yeah.
Only adults who are at least 18 years of age. There's not a hard upper limit. It's again something that should be looked at carefully, I think, in people who are perhaps over 70. But there it's not prescribed as a problem for entry.
Yeah. Okay.
And as you can see there's I think it was Gemma Smith asked what were the limiting comorbidities, and often these get worse as you get older, and they get worse if your Fabry's has been progressed some time. So all the severe problems of severe brain disease, heart disease, kidney disease, lung disease, things that would make it tough to go through any procedure. Currently in this first generation of gene therapies are exclusion criteria. If you've got active cancer, if you've got uncontrolled diabetes, if you've got chronic illness, chronic virus infections like hepatitis B or hepatitis C that haven't been suppressed. All those things may get worse when we damp down your immune system. But just from my impression as a clinician, these new waves of gene therapies are a huge advance on the first generation ones that required a stem cell transplant, which was a much tougher ask and was really reserved for the very young, the very fit classic males.
And then talking about that with the classic males, there's a question here. What's the indication that males with non-classic may be considered for the Sangamo phase 3? Michael, did you want to answer that one perhaps?
I'm actually not involved in the Sangamo trial. I'm just doing 4DMT at this time.
Okay.
But from the point of 4DMT, at least, it doesn't matter whether you're classic or non-classic. It just the criteria really around the clinical things. So, whether you have some disease involvement, but not too much, as Dr. Thomas mentioned. If you've got significant renal or cardiac problems, then you unfortunately won't make criteria at this time. So it's, it doesn't matter if you're classical or non-classical. Kathy might want to talk about Sangamo from that perspective.
I think it's all changing, and that each person, we should go through the situation for each individual person one-on-one, really.
That's wise.
Yeah, so there's not necessarily a classic, non-classic. It's about your level of your ability to participate based on your level of illness or fitness.
Yeah, the Sangamo has subgroups in it that are quite complicated.
And sometimes we set an arbitrary number. For example, if your heart ejection fraction is less than 45%, that means you're at least moderately severe heart contraction ability. But speaking as a kidney specialist, if it says your GFR has to be over 30 or something like that, I can tweak things. I think
40 is even 45, Mark. Yeah.
So although there may be a number there, if you're right on the border, come and have a chat.
Yeah, so I suppose the take-home message there is to just ask and touch base with your specialist and test that eligibility rather than assume one way or the other. Do you agree?
Absolutely.
Yeah, absolutely.
Yeah, absolutely.
So the next question: what's the expected time frame from trial to potential widespread treatment? Would you have any indication about that?
It's in terms of the trial itself, they're both open now for males, and we expect females to be open eligible within a couple of months, a month or two. So it's time. It's already time to talk about it with your specialist, really, if you're interested. Do you mean, if you mean,
so then how long are they earmarked for the trial program to continue? Do they have any idea about that yet?
No, it's going to depend on rates of recruitment overseas compared to us. There aren't any limits at this stage. The numbers involved in the Sangamo trial are probably going to be less than the 4D, but there isn't 4D have not prescribed an upper limit of numbers.
And then the difficulty, I'm not sure if you want to just explain a little bit about the process, then how these treatments from trial phase then to be listed on the LSDP.
Yeah, I think, that was part of the second part of the question, was it? I think we're a long way from that, to be honest, Sheridan. I think we're years away from this being standard therapy for Fabry. The field of gene therapy is moving rapidly, and there are now hundreds, if not thousands, of patients worldwide that have been dosed with gene therapy for different diseases, not necessarily Fabry. But, this is something that we really want to watch carefully because we don't really know what's going to happen in the long run. So that's part of careful clinical trial design and running these things properly. But it's going to take a long while, I think.
It's going to be a very expensive therapy.
Yeah. So it's a bit hard to, not predictable really at this stage.
Yeah.
A question here from Diane. My mutation is a deletion. Is this included?
Yes.
Yep.
Short answer, yes.
Does having gene therapy, this is an interesting one from Lynn, does having gene therapy exclude you from going back to ERT?
No, it doesn't. There are some of the preliminary studies overseas have kept people on enzyme and then taken them off when they're a few months in. We don't have that. we don't have that luxury because LSDP enzyme is not available to people on clinical trials. But it looks from the enzyme levels that you don't need it anyway, because the enzyme levels that people are getting with gene therapy are higher than the average levels on enzyme anyway.
I think this may be a little bit tricky, Kathy. I mean, this hasn't really been tested, hasn't it? Because if you're on a gene therapy trial, how long are you on that trial for? You're just on it for 12 months? Do you know what I mean? Like, I don't think we really know how the LSDP is going to interpret how all this means. So I would have a little bit of caution about saying, you know, you do the gene therapy, and then if it doesn't work, you can definitely go back onto LSDP drug 12 months later. I think you can, but I just don't think it's been necessarily tested yet. Is that fair to say, Kathy? What do you think?
Well, you can always, at that stage, you're not in any, not undergoing any active study.
Yeah.
You will be just monitored as part of the program, and it's the same with any clinical trial. If it's better for you to come off the clinical trial and not be monitored within the trial, then you're off the trial.
Yeah.
It's the paramount factor is what's best for the patient, of course, isn't it?
I've always found LSTP completely reasonable. You know, if you make a reasonable request, they'll absolutely entertain it. It's not a rigid, you fall here and you don't fall there. So
it's reassuring to hear, Mark.
Except
so
for
sorry, yeah.
No,
I see
that's
we've got a question here just regarding more protocols of testing. How often would the testing take place during the initial 12-month period and subsequent years? And how long would the testing period be? So that'll differ from one to the other, I'm assuming, but if you could just give us a bit of a ballpark kind of guideline.
Yeah, okay.
I can do that if you want. Do you want me to do it?
Thanks, Donna. Yeah.
Donna. So we'll introduce Donna now. Thanks, Donna, for jumping in. Donna is our Royal Melbourne Hospital clinical therapies nurse, clinical trials nurse, I should say.
Yeah.
Thanks, Donna.
That's okay. So after the initial infusion, it's pretty intensive for about 2 weeks. And then both of them pretty much have testing every week or two for the first sort of month. And then it gradually extends out to every 4 weeks, then to every 3 months. Both studies have some home visits once that gets approved at the local sites, which means that for very simple visits that only involve some blood tests, a nurse will come to the house if that's what you want. It's obviously optional, and collect the blood and send it off for us. And then at the moment, we don't have an extension information for Sangamo, but they will do a long-term follow-up. And we know that the official trial long-term follow-up for 4D at this stage is every 12 months after the first 12 months.
Okay, thank you, Donna. Thank you.
the cardiac biopsies are just on 2 occasions in the 4DM trial, so it's not a regular thing. That's not the test.
Long-term follow-up is very basic. It's very similar to what you do for the routine Fabry visits anyway.
So we've got a question here from a female who has heart disease. It's just wondering whether that makes her ineligible. So I think the answer to that would probably be to ask your specialist and discuss that to see whether you meet criteria. Does that sound like an accurate response to that?
Yeah, that's right. That's right. I mean, the answer is probably would be okay depending on what the actual numbers are.
Yeah.
But if it's Fabry heart disease, it actually gets you into the study.
Yep. So again, test the criteria and see whether you're eligible. Yep. Kathy, when Kathy says the trial may be open for eligible females soon, are there broad timeframes around this, 6-12 months? I think you indicated in the next few months, potentially, Kathy. 2 months. Thanks, Mark. Did you want to elaborate on that at all, Mark, or is this a simple answer?
It's the old race between the ethics committees.
Okay, so it's pretty much as soon as the ethics committees have organised, they'll be recruiting. So that's pending. It's imminent.
Yeah, Sydney, I'm really hoping to be enrolling before the end of the year.
Okay, so there's a question here from Sue. What is classified as classic Fabry disease? Now, I'll just briefly, from a patient perspective, but you can chip in. That's depending on your mutation. There are certain mutations that are considered classic, and then there's non-classic mutations. So, did you just want to quickly speak to that, Michael, in a nutshell?
Yeah, sure. So I guess classical Fabry disease, we consider patients who have, if we sort of separate them out, male and female, but a classic male is a man who has very low enzyme activity or no enzyme activity detectable at all. They tend to have high levels of lyso-Gb3, and they tend to have all of the other sort of textbook symptoms of Fabry disease, like the angiokeratomas and peripheral pain and gastrointestinal symptoms. And women that have similar symptoms fall into classical as well. Non-classical patients, the biggest group, I guess, are the cardiac Fabry patients who only really have manifestations in the heart. So that's very broad brush description. But
so often it's related to your mutation, though, isn't
that's
it,
right.
Michael?
That's right. Yeah. So some mutations we can very much predict whether a family will be classical or non-classical.
So the Sangamo trials, they are in Perth and Melbourne. Perth and Melbourne. Yep.
Not Perth. We're only 4D.
Okay, so just Melbourne. Okay, thank you. Thanks, Donna. Is there any data relating to the effect of gene therapies on neuropathic pain and the possibility of coming on Tegretol? Is there any early data we're talking about that?
Not as far as I know, but so if we extrapolate from our knowledge of enzyme replacement, then the levels of circulating enzyme that you get with gene therapy are likely to positively impact on pain, but will take a while, probably 12 months before we could answer that.
And then it's talking here, we've got another question saying the graph that you showed us, Kathy, had 40 weeks of results. Is that where we're at as far as data collection at the moment?
It's a little longer than that, but I can only show data that's been
published.
Published,
yeah. So that's the answer to that. We've got published data up until that point. So that's hopefully answering your question, Ben.
If you just
Deborah,
want to chip in about the neuropathic pain.
I'd love you to, Mark. Thank you.
The other question about pain is, particularly if you notice this when you first start the main enzyme replacement therapy, sometimes pain that is all burnt out comes back as nerves come back to life. So it can be tricky. It can go either way. And you think you're getting worse while you're actually treating and getting better.
Yeah. Does chaperone treatment make you ineligible for either trial?
No.
No. Lovely. So I'm just trying to move to the last screen. So I'm just trying to get back to my chat. Have we got more questions? Can anyone see the next question? Here we go. Sorry, I'm having a few tech issues here. Transplant patients excluded?
At this stage, I think it's
They
Yes,
are at this point.
Yes, they are.
From 4DMT, they are.
It's always the first stages of new treatments that go for that don't want to have transplant and dialysis patients in there. Over time, things come through for other groups of patients as well, but initially, the initial studies are always exclude transplant and dialysis patients.
Yeah. And I think as far as, here's another question from Stephen, I think as far as it's the best place is to ask and discuss it with your treating specialist who understands your case exactly, and then also you can consult with one of the specialists that are running the trials to see. So I think that's the answer there. Have we got any more questions down there? If you can see any more chat questions, just keep going.
I think you've answered everything that I've seen, Sheridan. Yeah.
Thanks, Di, because I just wasn't sure if my computer wasn't scrolling down properly. Here's one from Sarah Williams. Is this viewed as a cure? Interesting question.
Well, that's the hope, but I think it's
yeah.
I don't, we don't know.
Yeah, I guess I'm cautious about using the word cure. I think this is very likely to be much better than ERT. But I think there's a number of reasons why it's less likely to be a full cure, to be honest. And I won't go into the details, but I think there's a number of reasons why that, this is not going to make a person with Fabry disease like a person without Fabry disease.
Some of the degree to which it approaches a cure will depend on age at which you have the treatment.
Yeah,
and it doesn't get into your gonads, so you may have your own body cured, but you'll still pass on the disease to your future general unless you look at that fertility and selection.
Yeah. So the various genetic viral vectors that are used don't necessarily go to every cell of the body. So that's one of the issues. We don't know how long they will stay in the body. It's likely they'll stay for a very long time, but we can't guarantee they'll stay forever either. So there's a couple of the reasons why I would be cautious to say this is likely to be a cure.
Mm-hmm. Good, but certainly hopeful, isn't it? That it's really another therapy that is another option for patients, isn't it? Which is good. I think the more options we have and the more decisions that we can make, I think that's really positive for everybody, isn't it?
Yeah, and the numbers of people who will want to take it up are going to be small, I think. And
we'll learn as time goes on.
All right, well, I think if we don't have any more questions, I'll say thank you everyone for your time this evening. Thanks to Dr. Kathy Nichols and Dr. Mark Thomas and Dr. Michael Chan for joining us. We hope you found this evening informative and have had all your questions answered. I can't see any more popping up, so I'm assuming that's a yes. Just a reminder that you'll find useful links on our Fabry Australia website. And then also we'll have a recording of this presentation. So if you want to come back and view it again and go over that again, that's fine. Also a reminder to register for our upcoming virtual conference, which will be in October, on the 15th of October. We'll open registrations for that in the coming week. So thank you everyone for joining us again and enjoy the remainder of your evening.
Thanks everyone.
thanks, Sheridan.
Thank you, Sheridan.
Thanks everyone.
Thanks to Fabry Australia.
Yep.